afternoon. Very pleased to see there's still quite a few members here. This is the end of our session. I'd like to first of all thank the organizers for inviting me and my colleagues to finish off today's session with the Symposium on Herbal Medicine and Cancer. So I will start leading the talk on new therapeutic approaches to overcome resistant cancer, and I will specifically mention about the role of herbal medicine during my talk. I'm from the Western University of Health Sciences, which is located east, about 35 miles east of Los Angeles. For those of you who doesn't know where this university, it's a health science university only, with medicine, pharmacy, nursing, dentistry, etc., about 9 different health science disciplines. So it's not a very big university, but it's it's considered to be the most comprehensive health science university in the United States. That's the one. Just a quick picture, how it looks like. And this is a clinic building with pharmacy, and a patient could see the clinic, relatively new building. Now, today I'll be talking about, briefly, the major mechanisms of cancer chemotherapy resistance and ways to overcome the resistance, and some of the actions of the herbal medicine, which I will describe. And the second main point I'd like to introduce is a concept using evidence-based personalized medicine that could lead to optimal cancer chemotherapy. Well, why am I interested in this topic? I think we know that at the present time, cancer is a leading cause of death worldwide. In the United States, it's a second leading cause. But it's predicted that in 16 years, this will be the leading cause, surpassing the cardiovascular death, which is the #1 killer in the United States and part of the Western world. It has been well documented, and most people agree that cancer chemotherapy resistance is responsible for 90% or greater than 90% of metastatic cancer. One of the problems about the chemotherapy resistance is that there are multiple mechanisms of resistance which can develop. And when a patient is initiated with an effective anti-cancer drug, or started with chemotherapy, invariably the patient would develop new resistance after a while, sometimes very rapidly, sometimes takes a while. So at the end, there's no good drug therapy. Patients with metastatic cancer would usually die. And I think that's why it's a #1 killer in the world. And I think there's a lot of work needs to be done, a lot of research needs to be done. And I'm going to be talking about an approach, hopefully, it will prove to be very useful. Just very quickly review the mechanism of chemotherapy resistance. It can be due to cellular mechanism or tumor physiological mechanism. For the cellular mechanism, a number of well-known specific mechanisms have been isolated and known now, including decreased drug uptake, alterations to drug metabolism, increased drug efflux, modification of drug targets, enhanced DNA repair, inactivation of apoptotic proteins, and activation of anti-apoptotic proteins, and others. For the tumor physiologic mechanisms, these usually include intermittent and irregular perfusion, limited drug diffusion, hypoxia, acidic environment, adhesion-mediated resistance. And I won't discuss too much detail except to illustrate some of the points of the cellular mechanism to illustrate the need for a new strategy for treatment of resistant cancer. One of the well-documented mechanism, and the drug companies and academic institutions have spent billions of dollars on this mechanism-based drug therapy. And this is it's called P-glycoprotein drug efflux mechanism. When anti-cancer drug is administered to the patient, drug will usually go to the cancer cell and try to kill the cancer cell. But the cancer cells are very smart. It will express P-glycoprotein and excess P-glycoproteins. P-glycoprotein, for many drugs, they are substrates of P-glycoproteins, which are transporters. The transporter will transport the drug inside the cell to outside the cell, therefore resistant to the drug, anti-cancer drug developed. So one of the specific mechanism where we could overcome this particular resistance is adding P-glycoprotein inhibitor. By inhibiting this transporter, then anti-cancer drug could retain in the body and exert its anti-cancer effect. And as a matter of fact, many drug companies have developed very potent inhibitors as a very important target in anti-cancer chemotherapy. However, it has not worked very well so far. Another quick mechanism is the trail. Trail actually is a ligand. Trail stands for the TNF-alpha related apoptosis-induced ligand. It's a ligand that's actually in our body. When there is a cancer, our body develops automatic defense mechanism to kill the cancer. So the ligand in the body will be triggered and bind to the death receptors, and through several cascades, caspases, and so on, and result in apoptosis of cancer cell. That's our normal defense. However, with potent trails as well as endogenous trails in our body, with the expectation that this would occur in killing cancer cells, there are other mechanisms that actually the cancer cell initiates and result in survival of the cancer cells. And I won't get into too much detail. And the drug companies, many drug companies have, over the past 15 years, developed very potent trails or trail sensitizers, try to activate this. But at the end, no clinical efficacy has been demonstrated, both for the PGP inhibitors and the trails. And so that this well-documented mechanism to kill the cancer cell has failed after multi-billion dollars of research in the world. And there may be multiple reasons. One reason is that I've shown you these could be demonstrated very effectively in vitro and in animals, but there is a discordance between what we observe in vitro and in animals in terms of efficacy versus clinically how these drugs work in patients with actual cancer. So what can we do? How can we improve? Obviously, we need a lot more strategies because in vitro and animal models doesn't work too well. It has been estimated from a drug development perspective, maybe no more than 5% concordance in the in vitro animal data, you could predict the clinical efficacy. And one of the important reasons is that difficult to treat cancer is that whenever a new anti-cancer drug has been developed and given to the patient, it may work initially, but soon the resistance will develop. And this is pretty well understood and accepted these days, even though it's still quite theoretical. When the patient has tumor cell in the body, when we give a very effective anti-cancer drug, there will be cells that will be sensitive to the cancer drug, and these will, of course, die. However, in the body, the cancer cell, there are presence of initial resistant clones, which will further develop stem cells, and then also tumor-associated extracellular matrix causing resistance. In addition, there are remaining cancer cells which has genetic instability, and they will then grow, and they will outgrow and result in resistant clones and develop resistance. So therefore, initial anti-cancer drug will kill some of the cells, then the remaining cell will gradually develop resistance, and then patient will become, the drug will be no more effective and die. So our traditional approach of targeting the mechanism, a very clear mechanism, one mechanism at a time, clearly has not worked so far for cancer chemotherapy. We must utilize some new strategy. A new strategy that has been talked a lot, and I'm very interested in this, we are actively pursuing this as well, is evidence-based personalized medicine. Because while it's wonderful to have molecular genetic mechanism and target it and understand the targets and so on, oftentimes, so far, at least in the animal model and in vitro mechanistic approach, the concordance to the clinical efficacy is only about 5%. So that's not very good. However, if we can use patients' cancer cells and directly using evidence-based medicine to target the cancer cells from the individual patient, this could be more effective for each individual patient, because each individual patient, the cancer cell is different from another one. And once if we can do that, and if we have a broader spectrum of drugs, in addition to the conventional anti-cancer drug, which approved by, say, FDA or any regulatory agency, which the patient will already have tried, for example, if we could add other avenues, such as herbal medicine, then and if it works, then it can really expand our armamentarium against cancer. So we need both. They're able to grow individual cancer cells and then utilize a large of so-called bullets, different bullets to target this individual cancer cell and find out which is the best bullet, and then give the best bullet to the patient. So this is a personalized medicine, and it requires 3 main steps to my mind. First, we need to culture individual patient cancer cell, and there are 2 techniques which have been actively working on in the world. One is a circulating tumor cell, I think you all heard about. This is probably the simplest one. Basically, patients with cancer, especially metastatic cancer, there will be circulating tumor cells available. You just have to draw the blood sample. Average so far is 7.5 cc of blood, and you isolate the tumor cells, and then you grow the tumor cells, and then after the tumor cell is grown, then you screen different drugs target against the tumor cells from the patient, and once you find the most active drug and relatively safe for the normal cell, then you can administer that drug to the patient. One of the problems at this point about circulating tumor cell, it's being actively worked on throughout the world now, is that there is no good technique to grow the circulating tumor cell yet. The technique of isolating it is already pretty good now. A number of publications has been established successfully, but to grow it is still in the very early stage, and we are in collaborating with other cancer centers try to work actively in this area. And so I think many other institutions are doing it, and I think it's a matter of time we'll develop a good technique to isolate. The second technique is conditional reprogrammed cell. This is a little bit more mature, although still a lot of research needs to be done. My colleague, Dr. Liu from Georgetown University, is the one who pioneered and patented this particular technique. This requires a tissue biopsy sample obtained from patient with tumor, and then using special technique, I'll show you a little bit later, and able to grow the individual patient's cancer cell. And then we can, again, screen different anti-cancer agents to find out which is the best one to kill the individual patient cancer cell. So once we determine the individual drug, anti-cancer agent, or combination of agent that's most effective against the patient's cancer cell that's growing in vitro, then it's like antibiotics for microorganisms. Culture sensitivity, find out which is the most sensitive against this most sensitive agent or combination against the cancer cell. The next thing is to give to the patient. And that's the third step is individualization of dosage. As you know, especially many herbs, we don't know what's a safe dosage for the individual patient. However, there are new techniques already available widely used in China and other places using the LCMS and techniques and so on to look at active components, individualize dosage based on individual pharmacokinetics, and then give the right dosage. And this, so stepwise, these 3 steps, I believe is very much needed. Let me just expand a little bit on the condition reprogram cells. This, for those of you interested, these are the references originally published by Dr. Liu in 2012. And what he has done, basically, this particular technique using a ROCK inhibitor and feeder cells, there's a reprogram the cells and grow the cells. You could grow the normal cells and then transplant in vitro and transplant it in the mice, a xenograft model, there's no tumor showing there, or a carcinoma, a cancer cell is able to grow in 2D culture or 3D culture, and then transplant the cells into the mice and develop a tumor, xenograft technique, and this is very successful for prostate cancer. It's 100% success. And so this allows one then to determine culture sensitivity. Here's actually an example. This is, however, only one case example, but the case is so good, so convincing, it was published in New England Journal of Medicine. Only one single case. It's a proof of concept. This is about a patient who developed carcinoma of the lung that was initially triggered from the respiratory papillomatosis, which is very difficult to cure. And then Dr. Liu's group was able to culture the carcinoma of the lung once it developed, and then screen through multiple drugs. And this drug, obviously not so good. This drug is not so good. You can see here's the survival, 100% growth, and there's a concentration of the drug, and the cells would not be killed by the drug, as you can see by here. Verinostat, here's at the low concentration, 100% growth of the cancer cell, and then you can see as the concentration of this drug is increased, IC50 that kill 50% of the cancer cell. You can see is here. However, you can see that the IC50 to kill 50% of normal cell, the concentration is much higher. Therefore, this concentration could be a targeted concentration to kill the cancer cell, but not so toxic to the normal cell. And therefore, it can be implemented. And this actually is a patient that before the treatment, very large tumor, and then 3 months after treatment, tumor has shrunk. And then at this particular report, at 15 months, the efficacy was maintained. So it's a proof of concept, even though it's only one case, but I think it provides the personalized medicine approach is possible using such a technique. So we obviously would like to use such a technique and provide more effective therapy as well as combination, and how we can enhance our efficacy in implementing such an approach. I think here's where multiple drugs combination need to be tried, and herbal medicine can help. And let me show you, for example, this is our data that we have done, that this is a prostate cancer cell, is the resistant prostate cancer cell to docetaxel, which is the drug of choice for metastatic prostate cancer. And you can see the IC50 to kill 50% of this resistant cell, you need concentration around here. And this is, in contrast, this is a sensitive PC3 sensitive cancer cell. All you need is much less. So the resistant cell usually is 5 to 8 times higher concentration that's needed to kill. When we add a small amount of herbal medicine to the docetaxel, and we look at the docetaxel resistant prostate cancer cell, IC50 is returned back, shifted back to the sensitive cell, indicating that certain herbal medicine, when you combine with a Western drug that is resistant, you could regain its sensitivity. And this could be very exciting. Many herbal drugs can do that. I just want to show one example. We have done a number of drugs screened out. And just to show a little bit to prove that there was cell line model, and here's a model actually in animals where we look at the PC3 sensitive to docetaxel. When the tumor is grown, we give docetaxel. Docetaxel either maintained the tumor size or reduce the tumor size over time. However, in the resistant cells, resistant to docetaxel, when we induce a tumor in mice, and when we looked at the docetaxel treatment, these are the treatment days, and the tumor was continued to grow because of resistance. Therefore, we know we have a good model. So then we just illustrate the principle that docetaxel plus the Chinese medicine, the one I showed you earlier in the in vitro, here is a combination in this resistant, docetaxel-resistant tumor that's growing in the mice, and these are the other groups, docetaxel alone, control, or Chinese medicine alone, but the combination is the one that really worked, and just proof of a point. Again, this is animal in vivo data, so it's just to illustrate a principle, not in terms of clinical efficacy. Another very important role in expanding the chemotherapy ability is another effect that I want to illustrate. We call collateral sensitivity. And for the herbal medicine, I believe this is one of the first that we ever reported. This hasn't been published yet, but I wanted to show you. This is in vitro data in using the same cell line, the prostate cancer, trail resistant. Remember I told you about trail being endogenous substance, that if you give it endogenously or drug company isolated trail to give to patients, it's not clinically effective because resistance develop very rapidly. And so this is a resistant cell line that's resistant to trail, and this is a sensitive cell line, and this is a PC3 cancer cell resistant to docetaxel. There's 3 here show. This particular herbal medicine is a single compound isolated from the herb, extracted from herb, that in the resistance, here's a resistant, here's 100% growth, and here's the concentration of this herbal compound against each of these cancer cells. You can see in this particular case, trail-resistant cancer cell is able to kill 50% of that at this concentration, whereas for the other cancer cells, it's much higher. Okay. We didn't have the normal cells here, but the normal cells will be around here. So again, this particular herbal compound will be very effective alone against resistant cell without sensitization. So this is, we call collateral sensitivity against the resistant cell specifically. This is just another example. I won't spend the time. And the last part would be that we could look at a combination effect of different cells. Here's, again, the trail-resistant prostate cancer cell as an example, that if we give trail, this is in terms of concentrations, this is looking at 100% growth using this trail. If without the other one, if we combine with this herb, single extract, again, relatively high, almost 100% growth. If you combine with the other one, combination at the low concentration, again. But if we add this low three concentration together, it becomes effective. And then if we increase one of the concentration of the other one, it becomes more effective, indicating that herbal compounds plus the existing anti-cancer drugs, if you plan it well and combine it well, it can be very effective in resistant cancer. So basically, what I wanted to say is that we need a new strategy for treatment against, for the treatment of resistant cancer cell. And one of the strategies called collateral sensitivity approach, in addition to chemosensitizing approach. You need multiple anti-cancer activities to combine together to kill the cancer cell. And if we have two different anti-cancer cell evidence-based approach, then we can utilize this called collateral sensitivity cycling approach, which is shown here. Cycling approach is using two drugs which have against different resistant cells at different times. For example, drug A. Drug A is shown to be effective against certain cancer cells in the body. But if we continue with drug A, the resistant clones will be coming up, and drug A is no more effective. But then at that point, we hit it with drug B, which is, again, collateral sensitivity. Remember, when it's become resistant, we hit it with drug B. With collateral sensitivity, it will kill the cell. But if we continue with drug B, the resistance will develop, the heterogeneous nature of the cancer. So therefore, soon drug B will not be effective. But then we can treat it again with drug A after the time, the so-called cycling. Of course, we can use a different drug as well. So if we have this culture sensitivity approach, we can look at this at the time of drug A. We can culture the patient cells to see if drug A is effective. We give the drug. And when it becomes resistant, we could culture the patient cells again to see if the drug B is effective, and then we could give the drug to drug B. That would be more practical approach to monitor the patient's progress. Of course, in the meantime, this concept will help to kill the cancer cells as well as to prevent the development of resistance by cycling. This is very much similar to the current approach of treatment of antibiotic resistance, infections, and AIDS. You may remember in 80s, for those of you who are old enough, that when AIDS virus first developed and patient contracted AIDS, it's virtually fatal in a very short time. But now, with this kind of combination therapy, you can control AIDS. They don't have to die. Patients live quite normally, but you have to continue treat patient and monitor. And my thought would be, we could do that for cancer. We have to use this approach, and we could use evidence-based medicine approach. That's what we need to develop and work together. And I think there's a lot of work to be done. So in closing, I say in view of the heterogeneity of cancer cells, a personalized medicine approach to continuously monitor the efficacy of treatment in an individual patient should be a desirable approach for chemotherapy of resistant cancer. Expanding the armamentarium of anti-cancer agents with herbal medicine and utilizing new strategy to prevent the development of resistance, that is collateral sensitivity cycling approach, for example, could further improve efficacy, minimize the resistance. Further research, I hope collaboration among the researchers in the nation, internationally, to demonstrate the applicability of this concept is very desirable and hopefully would be very helpful in the future. Thank you for your attention. Okay, I will continue the second presentation for Dr. Ming Wong, who's the Dean of Sun Yat-sen University College of Pharmacy. Unfortunately, he cannot make it because of a very important large grant defense occurring at the same time. not want to lose the grant support, so he asked me to help out to present his slides, and I will do that. And that's also on herbal medicine that the title is Anti-Tumor Activity of Extracts Derived from Taxus Yunnensis and the Involved Pharmacokinetic Mechanism. Paclitaxel is one of the most important anti-cancer drugs widely used for the treatment of various solid tumors. And you can see the structure over there, and it's well known. The oral bioavailability of paclitaxel was significantly increased when co-administered with cyclosporin A, and that's been well known. However, such combination can result in toxicity. Now, certain TCM stand for traditional Chinese medicine, which is usually composed of herbs, extracts, and so on. TCM ingredients have been attracted a lot of attention, and it could be widely therapeutically effective and with relatively low toxicity. Some natural products were found to inhibit the activity of PGP, thereby enhancing the oral absorption of paclitaxel. So that could be one of the mechanisms that combination of the herbal medicine could be very useful. And here's some examples of TCM ingredients that enhance oral bioavailability of paclitaxel. You can see the combination resulting in higher concentration and area under the curve as compared to paclitaxel alone. Taxus yunnensis is endemic to China and has been used in traditional medicine for the treatment of cancer, diabetes, and other problems. Paclitaxel is the representative compound in Taxus yunnensis. There are, however, several other active components or ingredients. In addition to taxane type, there are biflavonoids, lignans, phenolic compounds, etc. So how about the anti-tumor activity of the extract from the twig and leaves of Taxus yunnensis? Is there any pharmacokinetic interaction that could provide its distinct advantage? So this part would be on the experiments using Taxus yunnensis on the A549 cell line xenograph in nude mice, and basically the nude mice was administered with the herb extract for 24 days, and then tumor volume, tumor weight, body weight, paclitaxel concentration in the tumor tissue were determined. And the result showed that the Taxus extract at the higher dosage, 480 milligram per kilogram, significantly reduced the tumor using the xenograph model compared to other regimens, including paclitaxel itself, 10 milligram per kilogram IP. This is the extract at that dosage is more effective against that paclitaxel. This is just showing over time, post days. It's the same information that I have shown you. And looking at the inhibitory rate in terms of effect, you can see that at that concentration, which is the most effective from the last one, 480 milligram per kilogram, equal to paclitaxel 10 milligram, it is 97%. So this particular dosage has paclitaxel equivalent, but it's more effective than this paclitaxel. And one of the other important points is that in this experiment, there's no significant difference. The weight of new mice of each group indicating the toxicity of Taxus urinaensis is relatively low, which is desirable in that case. This slide shows the concentration in terms of paclitaxel tumor concentration represented by different treatments that you can see. This is the herbal extract, which is the effective dosage, which has a very high tumor concentration compared to even paclitaxel IP alone, which is somewhat lower. But remember, they contain equal amount of paclitaxel. So obviously, there is some other ingredients in the extract that will enhance the concentration. And using various compounds and to determine, using LCMS, that one can determine concentration of various components that's contained in the extract, as well as paclitaxel. And you can see here, this very successfully analytical method that's been determined, and that contents of 7 toxoids are shown here in terms of paclitaxel. There's multiple concentrations in this extract itself, okay? That's just show additional other components using this particular assay. So the next study is on comparative pharmacokinetics of paclitaxel after oral administration of Taxus urinaensis extract and pure paclitaxel to rats. And the design is paclitaxel either 10 milligram or 30 milligram per kilogram oral dose, and then you compare to the Taxus urinaensis extract, which is equal to the 10 and 30 milligram PO in terms of content. This is the pharmacokinetics after oral administration. You can see that this is the paclitaxel, this is the herbal extract, equivalent to 10 milligram per kilogram. The extract resulting in much higher plasma concentration, and at the other dosage, also much higher. So basically, this is more detailed, looking at the 4 milligram per kilogram extract is equivalent to 22% compared to 10 milligram of paclitaxel, and the higher dosage is 18%. So it's just quantitatively telling you it's much higher. So there's a coexisting ingredients in the Taxus urinaensis extract has some special effect, must be on the PGP, and this is there were a number of compounds isolated from this herb, and then from the leaf and twig, and then they were fingerprinted and determined, and then we're looking at the using the MDR1, MDCK2 cell monolayer model to look at the changes in the PGP and the changes in terms of paclitaxel transport. What has happened is that certain of the ingredients that's in the extract has a great effect on the paclitaxel efflux. And I think I'll show you graphically in terms of time. Some other ones has no significant effect. And what's numerically summarized is here that the key components like this one has much greater inhibition effect and as well as this one. So these are the specific ingredients identified to enhance the concentration and to inhibit the efflux through the PGP inhibition. So this illustrates the kinetic mechanism and on the PGP, and which allow the herb to be more effective than paclitaxel alone at equivalent content, and further work in terms of mechanism, underlying anti-tumor activity of the extract and the formulation is in progress. I think that's the last slide. And that's the group from Dr. Ming Wang's group. So I'm just showing the slides, so I would encourage you that if you have individual questions, here's his email. I put that so you could contact him directly. Thank you very much for the attention.